DNA replication is the process of making an identical copy of a strand of DNA. Adenine is green, thymine is pink, uracil is later purple, guanine is blue, and cytosine is yellow. Colored ribbon is used for the sugar phosphate backbones. DNA replication begins in the nucleus. Helicase unwinds the parent double helix DNA strand. Molecules of single-strand binding proteins stabilize the unwound template strands at the replication fork. Primase begins synthesis on the leading strand. The leading strand is then synthesized continuously in the 5-3 direction by DNA polymerase III. Anti-parallel elongation, base pairs are matched through hydrogen bonds, adenine with thymine and guanine with cytosine. On the lagging strand, primase synthesizes an RNA primer at the 5' end of each Okazaki fragment. DNA polymerase III then elongates each Okazaki fragment and adds onto its primer. DNA ligase then joins the Okazaki fragments. The enzyme DNA polymerase I then removes the primer from the 5' end of each fragment and replaces it with DNA base pairs, adding on to the 3' end of the adjacent fragment. This process of DNA replication continues beyond the replication fork and occurs continuously until both the parental leading and lagging strands are completely replicated. Now both parental DNA strands have complementary strands.
RNA transcription. In the nucleus, RNA polymerase now binds to the promoter region of the new DNA strand and begins unwinding the two strands. As the RNA polymerase moves down the DNA strand in the 3' to 5' direction on the template strand, new complementary base pairs are added to the template strand, with uracil used instead of thymine. Once the polymerase finishes a section, the DNA strands begin to wind back up, and the RNA strand is detached from the leading DNA strand. Once the new mRNA is completed and released from the DNA template, a cap made of a modified guanine nucleotide is added to the 5' end, and a poly-A tail made up of adenine nucleotides is added to the 3' end to ensure that all coding parts of the RNA molecule are protected and sequenced to synthesize protein later on. RNA is processed so that the non-coding bases of the mRNA, called introns, are cut out, and the coding regions, called exons, are spliced back together. Translation. The completed mRNA then leaves the nucleus and travels to the cytoplasm. It binds to the large and small ribosomal subunits at its 5' end, and translation is initiated. RNA's nucleotide sequence forms 3-part codons, each of which translate to a specific amino acid. All protein molecules begin with the amino acid methionine, coded for by the sequence AUG, called the start codon. tRNA molecules are responsible for reading the mRNA codons and matching themselves with their attached amino acid according to their complementary anticodon. The initiator tRNA molecule binds to the mRNA at the P, or protein site of the ribosome. From there, additional tRNA molecules are added according to the mRNA sequence at the A, or amino acid site of the ribosome. The P site then becomes the location at which the most recent amino acid is transferred to the growing polypeptide chain located at the A site. The final site, the E or exit site, is where the empty tRNA molecule waits before being pushed back into the cytoplasm to be recycled with another amino acid. Translation continues along the mRNA molecule until a stop codon, either UAA, UAG, or UGA, is reached. At that point, a release factor protein binds to the mRNA and releases it from the ribosome. The ribosomal subunits, the mRNA, the tRNA, and the newly formed peptide chain are released into the cytoplasm to be broken down or made ready for use by the body.